Hello and welcome dear viewers to the another interesting video on scale up and post approval changes, that is also known as SUPAC, and this video is for SUPAC for immediate release formulations solid oral dosage forms. This is a part 1 of this video and I have referred a guidance for industry from US FDA, that is immediate release solid oral dosage forms scale up and post approval changes, which will involve chemistry, manufacturing and controls, in vitro dissolution testing and in vivo bioequivalence, and the documentation requirements. So as we have seen, the phases of drug development are research and development, which involve lab scale design and development, prototype formula and process. Then scale up and technology transfer is there, which involve product development and tech transfer, exhibit batches and process qualification batches. Then comes to the validation and commercial production, and the fourth is scale up and post approval changes. So after approval of the formulation and the commercial production, whether the commercial production is going on or not, after the approval, if the changes are done to the scale, that is scale up or scale down, and the other changes to the composition, manufacturing process, equipment, sites, those all comes into the SUPAC changes, and this is also is a part of life cycle approach for the product. So SUPAC changes involve changes to the components or composition of the formulation, changes to the site of manufacture, changes to the scale up, that is changes to the scale of the formulation, that is batch size. The batch size may be increased or decreased, and changes to the manufacturing. In the manufacturing, the changes may be for the process and the equipments. Then why SUPAC guidelines are there and why scale up and post approval changes are done? See, because the pilot batches or EB batches are with the small batch sizes, and the number of batches required will be more to meet the market requirements if the batch size is kept similar. Thus, the batch size is increased and the product is formulated for the commercial supplies. That time the scale up is done, and that's why the name is scale up and post approval changes. So other changes are also there, which will involve manufacturing at the additional site or additional manufacturing site to meet the market requirements, that is for the high volume products, and thus the site changes are required. Then the excipient supplier may stop producing the particular grade of the material, thus the grade cannot be made available, and so the composition change is done with the grade or another grade. For example, if the microcrystalline cellulose is available in certain particle size grade and the manufacturer stops that manufacturing, or other vendors are not there with the similar grade, then the composition is required to be changed. Then changes to the colors and flavors as per the market requirements, availability of the monograph or revisions of the monographs, the changes are required for the specifications, and this is very common practice into the life cycle approach for the pharmaceutical products. The old equipments may get replaced with the new ones, that's why changes to the site or batch size is required. Then changes to the process due to the cost considerations and also the patent's expiry. If the product is approved with the non-infringing technology and now the patent expired, so the old and tedious process can be changed to the simple process. That's why the process may change. Then sometimes the multiple changes are required to be done to the formulation in the life cycle of the formulation. That's why SUPAC is very important. Then SUPAC applicability. The SUPAC guidelines are applicable to the NDAs, that is innovator formulations or innovators, also to the generic applicants and to the abbreviated antibiotic applicants. So NDAs, ANDAs and AADAs. So all these categories the SUPAC guidelines are applicable. Then the SUPAC guideline defines the level of changes. Then it recommended chemistry, manufacturing and controls test for each level of the change. Then in vitro dissolution test and in vivo bioequivalence test for each level of change, and documentation that should support the change. So these are the important parameters or important aspects of the guidance. Then before going to ahead, we have to understand the basic definitions. Here is the definition of batch. Batch is a specific quantity of a drug or other material produced according to a single manufacturing order. So here it is single manufacturing order. If you take 100 kg granulation, then 100 kg will be the batch size. And during the same cycle of the manufacture and intended to have uniform character and quality within the specified limits. So a single batch is the quantity which is processed at a time with the single unit operation. Then contiguous campus, contiguous or unbroken site or a set of buildings in adjacent city blocks. So these definitions are very important when we will study the each level change, that is changes to the batch size, changes to the manufacturing site and all. Then dissolution testing. Dissolution testing, case A is dissolution of Q is equal to 85% in 15 minutes in 900 ml of 0.1 normal SCl is using the USP apparatus 1 at 100 revolutions or apparatus 2 at 50 rpm. So this is a standard dissolution condition. Then case B, multi-point dissolution profile in the application or compendial medium at 15, 30, 45, 60 and 120 minutes or until the asymptote is reached for the proposed and currently accepted formulation. So this is the case for multi-point dissolution profile. Then case C for dissolution testing. It involves multi-point dissolution profiles performed in water, 0.1 normal SCl and USP buffer media at pH 4.5, 6.5 and 7.5. So 5 separate profiles for the proposed and currently accepted formulations. That means the formulation without change and formulation with change, these formulations are required to be tested as per the case C for dissolution testing. Adequate sampling should be performed at 15 minutes, 30 minutes, 45 minutes, 60 minutes and 120 minutes until either 90% of the drug from the drug product is dissolved or asymptote is reached. Asymptote means then the dissolution is stopped. Suppose you get 84% release at 60 minutes and also 84% release at 120 minutes. That means the asymptote is reached. And now the more time will not give the higher release of the drug. A surfactant may be used with the appropriate justification. Then the drug product. What is the meaning of drug product? Drug product simply is a finished dosage form. That may be tablet, capsule, solution, suspension, anything that contains the drug substance generally, but not necessarily in association with one or other ingredients. A solid oral dosage form includes tablets, chewable tablets, capsules, and soft gelatin capsules. So the drug product is simply is a finished formulation. Drug substance is an active ingredient that is intended to furnish the pharmacological activity or other direct effect on the diagnosis, cure, mitigation, treatment or prevention of the disease or to affect the structure of any function of the human body, but does not include intermediates used in the synthesis of such ingredients. In these drug substance definitions, we should not include the intermediates. Then coming to the equipment. Equipment may be automated or not automated, mechanical or non-mechanical equipment used to produce the drug product, including the equipment used to pack the drug product. So equipment involves all the machineries. Then formulation is a listing of ingredients and composition of the dosage form. Justification means the report containing scientific data and expert professional judgment to substantiate the decisions. So this is the simple meaning of justification. New drug substance. It is a drug substance or any substance that when used in the manufacturing, processing or packing of drug causes that the drug to be new drug, but does not include the intermediates used in the synthesis of such drug substances. New drug substance simply means the new APIs. Then operating principle. These are the rules or concept governing the operation of the system. For example, you coat the pellets by Brewster coating. So Brewster coating will be the operating principle. Then pilot scale. The manufacture of either the drug substance or drug product by procedure fully representative or simulating that used for full manufacturing scale. So the process will be same as per the commercial manufacturing process. For solid oral dosage form, this is generally taken to be at a minimum. So this is a pilot scale. 1/10 of the production batch. If the production will be 10 lakhs tablet, the 1/10 of that full production will be 1 lakh. or 1 lakh tablet or capsules, whichever is larger. If production batch size is 20 lakhs, then 1/10 of the 20 lakhs will become 2 lakhs, and that should be the batch size. Now, process, a series of the operations and the actions used to produce the desired results. Ranges are the extent to which the limits between the acceptable variation exist. Same means agreeing in kind, amount, unchanged in character or condition, and similar means having a general likeliness. So these are the important definitions. Then definitions of scaled up is simple. It is the process of increasing the batch size, and scaled down means the process of decreasing the batch size. Then coming to very important parameter, that is significant body of information. So a significant body of information means the body of information on stability of the drug product is likely to exist after 5 years. So if the production is going on for 5 years and stability data is generated, then we can say that the product is having the significant body of the information. Or commercial experience of new molecular entities, or 3 years of commercial experience of new dosage forms. So that is the meaning of the significant body of information. Then validation. All of we know that validation is the establishing through the documented evidence that our process is capable to consistently produce the desired quality product. So its definition is establishing through the documented evidence a high degree of assurance that specific process will consistently produce a product that meets its predetermined specifications and quality attributes. A validated manufacturing process is one that has been proven to do what it purports or it is represented to do. The proof of validation is obtained through the collection and evaluation of data, preferably beginning from the process development phase and continuing through the production phase. Validation includes process qualification, the qualification of the material, equipment, systems, buildings and personnel, but also includes the control of the entire processes for repeated batches or runs. So this is the very important terminology of the validation. Then coming to the important part of this guideline, changes to the components and composition. In this video we are only going to study in depth the changes to the components and composition. So what are the levels of changes? Level 1 change, level 2 change and level 3 change. So as the number is increasing, the changes are going to have impact onto the product quality attributes, and this impact is to the quality and the performance, and it involves stability, dissolution, that is in vitro and in vivo performance. So chemistry, manufacturing and control related information is required to be given if the changes are more or if the changes are likely to have impact. Coming to level 1 change, unlikely to have any detectable impact on formulation, quality and performance. Level 2 changes could have significant impact, and level 3 changes are likely to have significant impact. So level 1 changes are unlikely to have detectable impact. The example is deletion or partial deletion of an ingredient intended to affect the color, flavor of the drug product, or change in the ingredient of the printing into another approved ingredient. So level 1 changes are very common changes, and these are unlikely to have any detectable impact, like you have changed the color of the film coating, or you have changed the color of the capsule shell, or you have added the printing ink, or you started the printing the tablets or printing on the capsule shells. So it will have no impact on the formulation performance. Then changes in the excipients expressed as percentage weight by weight of total formulations less than or equal to the following percentage ranges. So here the percentage ranges are given. For filler or diluent it is 5%, disintegrant starch is 3% plus minus, other are plus minus 1%, for binder it is 0.5%, lubricant if it is calcium or magnesium stearate 0.25%, and other it is 1%, lightant if it is talc 1%, and others 0.1%, and film coats it is 1%. So the cumulative changes or the total additive effect of all excipient changes should not be more than 5%. So for example, if you change starch around 3%, that means you increase the starch 3% and you decrease the quantity of this 3% starch with filler and you decrease the 3%. So total addition will be 6% and not the 3%. The total addition or cumulative or additive effect of all excipient changes will be 6%. So it will cross the limit of 5%. All the components should have numerical targets, and these allowable changes in the composition should be based on the approved target composition and not the previous level 1 changes in the composition. So if you are doing a change at second time, that time you are not supposed to compare the formulation being changed as second time with the first change composition. It should be compared with the original approved composition only. Now, what are the requirements of test documentation if level 1 changes are done to the formulation? So, chemistry documentation, application compendial release requirements and stability testing is required. Stability testing involves one batch on long-term stability and data reported in the annual report. See, this annual report is generated in the production plant, and every product is documented for the annual report. Then dissolution documentation, none beyond the application or compendial requirements. So here in the chemistry documentation is also same, and in dissolution it is also same. No additional dissolution data is required. Only the product release requirement is there, and the product is required to be stored on long-term stability only, and data is required to be reported into the annual report. Then in vivo bioequivalence is not required. Filing documentation is required in the annual report. All information including the long-term stability data. Then coming to the level 2 changes. These level 2 changes could have significant impact on the formulation quality and performance. And the test and the filing documents requirements depends on these three factors, that is therapeutic range, whether the API is narrow or non-narrow therapeutic index, solubility is high or low, and permeability is low or high. That means these three criteria came into the picture for level 2 changes. First is therapeutic range. The product is narrow therapeutic or not. The API is having solubility of low or high and permeability low or high. So based on this, the level 2 changes requirements are there. The example is change in the technical grade of excipient. Example is AVCL 102 versus pH 200. Changes into the excipient expressed as percent weight by weight of the total formulation greater than those listed above for level 1 change but less than or equal to the following percentage ranges. So if your formulation is going to be changed with the more than 5%, then it will come to the level 2 change. Please understand these things. The level 1 change is only up to the 5% change, that is total additive impact is 5% only, and if the formulation crosses that limit, that means if it is coming to the 6% or 7%, then the requirement will be for level 2 changes. The level 2 changes are enlisted here. Now the filler will have 10%, disintegrant starch 6%, others 2%, binder 1%. So these all the ranges are increased now, and the total additive effect of excipient changes should not change by more than 10%. So in level 1 it was 5% and level 2 it is 10%. And similarly, all the components in the formulation should have numerical targets that represent the nominal composition of the product on which any future changes in the composition of the product to be based. Allowable changes in the composition should be based on the approved target composition and not onto the composition based on the previous level 1 or level 2 changes. So whenever you are going to change the formulation first time, second time, third time, that time please refer the original composition and not the change one. Then coming to the chemistry documentation, application, compendial release requirements and batch records, stability testing, one batch with 3 month accelerated stability data, in supplement and one batch on long-term stability. So here now the requirements are getting increased. Earlier only one batch long-term stability data was required for level 1. Now it is one batch with 3-month accelerated data and one batch on long-term stability. Dissolution requirements are also changed. Case A, that is high permeability, high solubility drugs. You may refer these as BCS 1 or 3. BCS 1 only, because high solubility and high permeability is there. The dissolution of 85% in 15 minutes with the standard dissolution conditions. Then second, low permeability and high solubility. This might be the BCS class 3. Multi-point dissolution profile will be required, and dissolution testing is required to be done at 15 minutes, 30, 45, 60 and 120 minutes. And dissolution profile of the proposed and currently used formulations should be similar. So before change and after change, these formulations should meet the dissolution criteria. Then case C, high permeability, low solubility drugs. This is the case of class 2 drugs. Multi-point dissolution is required in water 0.1 normal HCl and USP buffer media at pH 4.5, 6.5 and 7.5. So separate profiles will be there. These will be 5 profiles and other things are the same. The dissolution profile of the proposed and currently used formulation should be similar. Then even have a bioequivalence requirements. There is no requirement if the situation does not meet the description in case A, B or C, refer to the level 3 changes. If it is meeting, then no requirement of bioequivalence. If it is not meeting, then it will go to level 3 changes. Then filing documentation. Here the prior approval supplement, that is PAS, is required with all the accelerated stability data, annual report containing the long-term stability data. Then level 3 changes, these are the highest level of changes. And if the changes in the composition are above 10%, then it will go to level 3%. It also depends on the therapeutic range, whether it is narrow therapeutic index drug or non-narrow therapeutic index drug. Solubility is low or high and permeability is low or high. Any qualitative and quantitative excipient changes to a narrow therapeutic drug beyond the ranges of 5%. So if the drug is narrow therapeutic drug and if the composition changes are more than 5%, then it will go to level 3 change. All the drugs not meeting the dissolution criteria of dissolution of 85% in 15 minutes. Changes in the excipient ranges of low solubility, low permeability drugs beyond 5%. Changes in the excipient ranges of all drugs beyond 10%. If any change is there and it is beyond 10%, then it will come to level 3. Chemistry documentation required is application, compendial release requirement and batch records. Here batch records are required. Significant body of information. That means the stability data of the commercial production batches, if it is available. One batch with 3-month accelerated stability data is reported in supplement. One batch on long-term stability data reported in the annual report. And if it is not available, that means significant body of information is not available, then up to 3 batches with 3-month accelerated stability data is reported in the supplement. That means prior approval supplement. And prior approval supplement means we have to make a document and give it to the regulatory authority and ask for the approval for the change. And then it will, if it is approved, then we can change, otherwise we cannot change. That is the simple meaning of prior approval supplement. And in annual report, we are not waiting for the agency's approval. We are directly doing the change and reporting into the annual report. So here up to three batches with 3 months accelerated stability data is reported into the supplement, that is prior approval supplement, and one batch on long-term stability data is reported in the annual report. Then test and filing documentation required. Dissolution documentation. Low permeability, high solubility drugs. Multi-point dissolution profile should be performed in application. Compendial medium at these 15 to 120 minutes, and dissolution should be similar. Then in vivo bioequivalence documentation. Here full bioequivalence study is required. It is very much important for the level 3 change. Once you go to the level 3 change, then always remember bioequivalence study will be required. And bioequivalence study can be waived only if the applicant has the in vivo in vitro correlation data. that is IVIVC data. Then filing documentation, prior approval supplement is must with all information including the accelerated stability data. Annual report will include the long-term stability data, because this long-term stability data will be generated after the approval of the prior approval supplement. So these very important terminologies are there, and interviewers always ask these terminologies. What is annual report? What is prior approval supplements? And what are the importance of PAS in the life cycle of the product? Then in this video we have understood the SUPAC's basic definitions, why SUPAC came, what are the SUPAC guidelines, and changes to the components and composition. In the next video we will discuss the site changes, changes in the batch size, manufacturing, in vitro dissolution, and in vivo bioequivalence studies as per the SUPAC guidelines. So you might have got a good understanding of this video. Do like, share and subscribe to the Pharma in-depth channel, and please keep watching the videos. Thank you.